Ok, parfait.
When you go to a meeting, you're just listening to very different science, neuroscience immunologists, or even further away. I find that really interesting, but then often I come back to my own research and see how it could connect. It's kind of making creativity through making connections.
Welcome to the Night Science podcast,
where we explore the untold story of the scientific creative process.
We are your hosts.
I'm Itai Yanai.
And I am Martin Lurcher. Ruth Lehmann is a professor of biology at MIT, and she's also the director of the Whitehead Institute for Biomedical Research in Cambridge, Massachusetts. For her many contributions to science, Ruth has been a member of the National Academy of Sciences, one of the most prestigious honorary organizations for scientists in the nation, since 2005. And she's also a Howard Hughes Medical Institute investigator since 1990.
Ruth's research focuses on germ cells and embryogenesis. And the germ cells, of course, are those cells that are the only ones in the human body that escape the fate of all the others, which is to just end with the individual. And instead, the germ cells, via the egg and sperm, they maintain immortality, essentially. So her lab studies the biological origins of the germ cells, how they transmit their potential to form a completely new organism. And she asks, what makes them so different? How do they interact with the non-germ cells so that they're protected? And so I was actually a colleague of Ruth's at NYU when she was the director of the Skirball Institute. And while we were both colleagues, I had a front row seat at viewing Ruth's tremendous role as a scientist. I really believe that she's a role model for not only what it means to be a scientist, but also what it means to be a leader that promotes how we do creative science. So, Ruth, we're so lucky that you're joining us today. Welcome.
Thank you. Thank you, Martin and Itai. I'm flattered. Thank you very, very much. It's great to talk to you.
No, it's fantastic to have you here on the podcast. Ruth, from your own perspective, can you tell us what the creative life of a scientist is like?
Yeah, I think if it wasn't for being creative, it wouldn't be much fun, right? Because a lot of the life of a scientist you could take like, oh, we have to write grants and you have to do this and you have to do that. And if it wasn't for the creative part, I always tell people when I get up in the morning, I know that I can determine what I want to do. And that is the most creative thing, right? That you are sort of self-empowered. And I often compare it, and I think you have done this in other podcasts, compare the life of a scientist with the life of an artist. And just like artists, and maybe even more tangible, some scientists contribute also to really improving health, improving many aspects of the world. But I do really see it very much like following your ideas. And that is just so exciting. It's the self-determination of being a scientist, which I think is wonderful.
Right. There is this sort of freedom, and it's the same kind of freedom that artists have. And I think our central question for you today, I think we're going to explore it in different ways, but when it comes down to it, the thing we really want to learn from you is, what is your process? What is your method for getting at creative ideas?
Yeah, I really like that you are putting sort of the finger on how you actually do it, because often I feel like it's something you just do. You don't think so much about, right?
Right. It's usually in the dark. We don't talk about it.
Yeah. I don't know if it's in the dark. Actually, for me, it's almost like that's the fun aspect. I would say that's the first aspect. And so just thinking about what means creativity or where does it come from? And to me, there are two really important aspects. And one is observing and the other is listening. And so the observing is that I'm a developmental biologist and I'm also a geneticist and I'm a cell biologist. So I kind of feel like there's these three disciplines, but I feel like where a lot of the ideas come from is I look at something and it may be a bunch of embryos under a microscope or a cross, and something is puzzling. something is not the way I thought it should be, or I'm observing something which I hadn't seen before. And obviously, the deeper you understand a system, the more you look at it, the more you can see these differences. And so to me, I would say the creative part doesn't come from, I don't know, sitting at the beach and having deep thoughts, but it's more just letting myself learn about my organism. And in particular, when you see something which isn't obvious or which isn't according to what you thought how it should be. I find these puzzles are the most intriguing and, of course, amazing when you can solve them.
Yeah, so it sounds like you're more an explorer than a philosopher.
Yeah, probably.
So what you just said was, at least as far as I understood, that creativity comes from noticing some contradiction or disagreement between the data that you have and your expectations.
Yeah, not necessarily even the data or just how you've seen it, right? It doesn't have to be necessarily, but it could be the data. I mean, I think what I'm saying is actually very similar, when Barbara McClintock, feeling for the organism, right? It's sort of the same thing. You really try to understand the organism and you understand it and with all its idiosyncrasies. And sometimes those idiosyncrasies are that make you question that it can't be just that simple as you thought it was.
So by a feeling for the organism, you mean you sort of have a mental image, a kind of model of how things should be. And then when you see some pattern in the data, that when you see something that shouldn't be there, that leads you to think of what could that be? Maybe that's something new.
So you said that it doesn't have to be in data, but I didn't quite understand what you meant. Can you tell us a bit more about that?
I meant more that the initial process isn't based on statistics. Let's put it that way. It's more the observation because you can't even put it into numbers at that point yet. Of course, you could say everything you see in any experiment is data, but I felt more like it wasn't grasped in a quantitative manner yet.
Yeah. No, that's very interesting. Actually, the way I meant it when I used the term data was just some kind of observation. It doesn't have to be quantitative. But it's interesting that you make this distinction, right? That you say your creative process is initially typically more concerned maybe with qualitative observations. Could you say it that way?
Yeah, I think that's right. Although I was just thinking sometimes when it's in genetics, right? You sort your flies and you figure out, you say like, oh my God, why do I get so many fewer of a particular number? Then it's obviously a quantitative. And so this is actually a nice example where you could say, oh, you get the wrong ratios. Those not the ratios you expected. So what could be the reason? That's actually a way of making you think.
Ruth, Martin's been asking you about the quantitative aspect, but I'm wondering if we could also flip the coin and look at the more emotional aspect. I think one of the things that scientists find particularly addicting is this thrill of discovery, these like aha moments and I think it would be a lot of fun to listen to you tell us about one such discovery, maybe one relating to your discovery relating to the gene Oscar or anything that comes to mind. We would love to hear about it.
Yeah, no, there's quite a few. Obviously, yeah, I mean, with Oscar, that goes very, very far back. That's very old. And I had many different kinds of experiments, and they're too complicated to talk about because there are all these transplantation and genetic experiments, which suggested to me that it seemed like Oscar was probably the most important or the critical or key gene which started this cascade of leading to nanos being active at the posterior pole. And to some extent, I don't know, is sort of a way to stratify your experiments because you have lots of genes you could be working on. I worked on Oscar because it had the most consistent phenotype too, and that was really important. And then it was the experiment of mislocalizing Oscar RNA. When we cloned it, we found the RNA was localized. We cloned it, put it to the anterior pole, and there it created everything, including localizing nanos there. And so that was very satisfying. But it was really in a way, that experiment that lots of people say, oh, that's a really important experiment. I always have a really hard time with this. What is creative? It's, to me, it's trial and error. It's letting, sort of doing, I often call this the Saturday afternoon experiments, to do an experiment which, you wouldn't normally do, but it's a very high risk experiment. But if it works, it would be wonderful. And so a lot of this led to a pretty good picture in my mind of how the system was working.
So what I found intriguing in your description was the Saturday afternoon experiments, right? These experiments that to me sounded like, okay, we don't need to do it, but it could be really fun and interesting. So let's just try it out. Is that what it was?
Yeah, and actually that is, if you ask me, how do you evoke creativity in my students, I say, maybe that's more a Saturday afternoon experiment. And then they look at me. And then eventually they know what I mean. It's not that you have to do it on a Saturday afternoon, but it also means that if it doesn't work, don't spend too much time on it afterwards. It's not like a one-time try, but you should leave yourself the possibility to explore. I always find that really, really important for my lab, that, they should sometimes, if there is something, if something catches their eye, and that's again coming from what I was telling you before.
Yeah. In a sense, with a Saturday afternoon, like you say, it doesn't have to be the day Saturday. It's kind of like an attitude. And Martin and I, we think about the notion of night science as not something that needs to be done at night. It's an attitude.
The night kind of seems to me like I get, I'm tired. But Saturday is this day where, it's not yet Sunday. It's a day of opportunity because you kind of have that freedom. And so that's what that sort of, chance to do something that is very self-directed and doesn't need specific, we often talk about, in a way, when people come to my lab, I say, and it's a little harsh, but the bread and butter project, which is very good because I think it's nice to have a bread and butter project, which means it's a project that from A to B to C. So, for example, in old days, it would have been, let's clone a gene, or, there are certain experiments, and they have to be done, and they're really important. And it also gives you a mission. I think also for a young student, it gives that kind of feeling of accomplishment because it's clear. And then you have this other experiment, which is more far out there and could work, could be very enticing. That's kind of what you're thinking about a lot. And so I like the balance because I think if you only did night science or Saturday afternoon science, I think it could be very frustrating too.
Yeah, potentially. I mean, at least there's a high risk, right? That it's not going to lead to anything.
It's a high risk, right?
I mean, that's one of the things about creativity. It's fun to be creative, but not every time you're creative, it's going to be something you can publish. So you need to find some balance there. So your Saturday afternoon experiments, do you maybe want to give us an example of, how you got maybe some wild idea for one of these experiments and how that influenced the project?
So it's sort of sometimes this question of asking, do we really understand completely how the system works? And if we did, then we should recapitulate it. And in our case, where we're looking at the entire embryo, it is always this question, or we're looking at germ cells, how they're formed. It is always the question of, can we then do that somewhere else? So it's not like recapitulating it in the test tube. And we've actually tried a lot of doing things in the test tube and they haven't worked as well and so we always come back to the embryo, and that's why we do a lot of imaging analysis, because we're trying to basically do biochemistry within the embryos by really quantitative. But a test for us is often, can we actually recapitulate? Do we know all the components? And this comes kind of back to the Oscar experiment, right? In the case of Oscar, if Oscar is really the gene whose RNA produces a protein, which then is the starting point for making germplasm, then if I put it somewhere else, then it should be sufficient to do that. So for me, often it's the necessary sufficient question. And so lately we have been talking about that, if we can actually take everything we know about how the germ cell itself is formed, which is completely differently formed in the embryo from the rest of the somatic part of the embryo, how that's formed. And then we had like lots of different components we would have to mislocalize, and that was kind of the experiment. So that would be an example of that, where we're saying like, oh, do we actually have enough of those components, and how could we manipulate it, and could we use combination of optogenetics, mislocalization. Became a really involved, and probably, I don't know, maybe it can work, but it became a really kind of interesting way of how all the thoughts we had of the process we tried to put together and think about how to do this.
Wow, Ruth. So as I'm listening to you, I can kind of imagine what your lab meetings are like. Tell me if I have it right. It seems as though you all describe the system as you understand it and try to think of experiments that either support or maybe in a surprising kind of twist would reject something that you thought you understood. I think that's a beautiful method. Is that what you're saying?
Yeah, definitely. I mean, I'm definitely of the part, like, I'm always talking about the killer experiment.
Yeah, spell it out for us. What would be a killer experiment?
Yeah, so the killer experiment is the experiment which would kill your favorite hypothesis. So I hate people who do correlative work. You found something and then you just do something which is obviously has to be the next thing and which would also be true, just to support their pet idea. I really like to do the experiment that really challenges my pet idea. Really back to the very classical ways of thinking of the experimental method, right? But this also often helps when you feel like maybe a project is sort of not moving forward, and it's really hard, and it sometimes is very hard when you have invested quite a bit of time in a project. Then it's also a good idea to say, so what experiment would really decide between life and death of this project?
All right. That's so interesting because I think in popular culture, I mean, like scientific culture, when people say killer experiment, they mean, I think, like such a cool
Oh,
experiment, such,
yeah. So maybe
so
my
amazing.
wording is the wrong. I should kill it. Yeah,
No,
I
no, but
take it in
no, I think
the.
yours is the right definition. I think that
Yes.
the one that really subjects your notion to the test, that's what we should really call the killer experiment.
No, it can be that public, because if it then supports your hypothesis, right? Then it is actually also in the general how it's often used. Then it is a killer experiment in that
Yeah.
way too, right? But I'm actually taking it more literally, that it actually could also kill the project, right?
Well, I propose we adopt your definition.
Yeah, so Itai was speculating about how your lab meetings go, but that's an interesting question to me. When you're being creative, is that typically something that you would do in a setting like a lab meeting or in a discussion with people in your lab? Or is that something, like an idea you have in the shower at the morning or on those Saturday afternoons where you're not in the lab?
I think it's all of that. It kind of come up in lab meeting, and I would say my goal is always that we do something that others are not doing and that is new and pushing us in a new direction. And so that often comes up at a lab meeting, for example. So, how much is that really pushing our knowledge forward? So that could be the discussion in the lab meeting. I have meetings one-on-one with people in my lab, and some of these, I know they talk among each other. If they want to have a lot of time with me, they know exactly when to schedule it and when they don't want to have so much time. And so it's like usually like three people in a row, So the last one, it can happen, that we talk for 3 hours. And that's because we get off on, oh, what about this? And that's often when these Saturday afternoon experiments come out. And then there are a couple of other ways. That's actually sometimes where I feel like we're thinking very deeply, but in a, we're not thinking about every experiment, but we're thinking really about the project as a large. Where do we want to go? What is really interesting, where we want to push this? So it's not about the day-to-day experiments. And so that's where that can go. We also have these retreats where the lab comes together and everyone talks about their projects. And what's nice there is sometimes projects which seem to be distant, they come actually together. And when things like, again, coming up with an idea which otherwise we wouldn't have come up. For me, the shower and walks, maybe more walks than the shower. I take very quick showers. Uh, but walks, I really like hiking. But what I do there is more when it's already, coming back to that observation that we talked about, when there is an observation that I find puzzling, I really think about that. And I don't know if you would call that creative or just really thinking deeply. And I feel like I have to have quiet and I have to block out everything for doing that.
That's really interesting. I think of the things you've said, it seems there's a pattern. When there's a difficult problem, it just simply needs more time. So your students, if there's a difficult problem, then they're going to need a longer meeting with you. And if there's a deeper pattern that demands an explanation, you're going to need a long walk to work through it. So I think there's kind of like this theme of persistence. And I'm wondering, what's your sort of mental habit for not becoming depressed or anything like that?
Yeah, what I find very difficult, and the more I've become, also involved in other things, like being the director of Skirball Institute, now being the director of the Whitehead Institute, where there's a lot of, that's actually kind of this kind of busy work. And it makes it very hard to get the time then to really deeply. So that's why other people who don't have the administrative work, and I used to do this, they have more time to just walk into the lab and chat with people. I just don't have that time anymore. And so meeting with people in my lab is really important for me to do that. Because we have also relatively frequent group meetings, it's really that time one-on-one is sort of different. It's really more thinking about the project deeper. And then to take me completely out, I really need that. I need to be out because otherwise there are going to be emails on my computer and calls. So taking the time and time on walks is just very, very important, or even long car drives, or just being not able to do anything else. I must have to prevent myself.
So do you feel that it helps if you're, like you said, if you're actively prevented from doing something else? What I mean is, I think this is true for a lot of us, that we don't give ourselves the license to just be creative. We always feel that we have to do something, and it's only in certain situations that we suddenly find ourselves in a situation that gives us the freedom to follow that creativity. Is that what you mean?
Yeah, and it's almost worse because you feel like, are you wasting time? Shouldn't you do something which has an outcome, right?
Yes.
So I think saying, no, this is not wasted because it's really important. But of course, you only realize that it's important when you do come up with a good idea. I mean, for example, I also like, this is one of the other, when you were asking me at the very beginning where creativity came from, and I said, it's a lot from observing, feeling the organism, but the other part's also listening. So I really love diversity of science. And so we really try to do that at Skirball Institute with having people from very different disciplines in the same institute. And I find it very stimulating to hear other science. And I find actually scientific conferences where, the best is, of course, these small conferences where people from all directions come. There used to be this Gordon conference called Biological Regulatory Mechanisms. And it was a conference which was everything. There were bacteria and plants and, everything was there. I mean, biological regulatory mechanisms, right? It's everything. And it really depended on the organizer. And this was one of my favorite Gordon conferences. It doesn't exist anymore because now everybody wants to go to their specialized meeting. And I feel when I go to a specialized meeting, I hear that one piece of data which just fits into the puzzle maybe, or I'm hearing, oh, somebody else is working on, problem we're working on, or I'm getting some reagent, that's not exciting, right? That's just like, yeah, you have to do that. That's kind of, I would put that in the bread and butter category. But when you go to a meeting, it's really, you're just listening to very different science, neuroscience immunologists, or even further away. I find that really interesting, but then often I always come back to my own research and see how it could connect. And that is definitely another aspect of, I don't know if you call it creativity. It's kind of making creativity through making connections, maybe.
Yeah, absolutely. Martin and I wrote a piece about that called Renaissance Minds, about the importance of interdisciplinarity. But Ruth, I have to ask you a question that is kind of different from the previous ones. You mentioned that now that you have leadership responsibilities, you're the director of the Whitehead Institute at MIT, and for many years, you were also the director of the Skirball Institute at NYU Langone, of course. And that makes me want to ask you about how you, as a leader who is also a scientist, how do you see your role as a leader in safeguarding science? I think that there's a logic to having our leaders in science be scientists themselves because they sort of get it. They know what science requires. And I had the privilege, because you invited me, to join one of the Skirball retreats. And I saw the culture that you established at the Skirball, and I was just very excited about it. I just saw many, many people genuinely excited about science, and that was so inspiring. So I'm wondering if you can just talk about what it's like to be a leader and how do you do it?
Yeah, I have given this a lot of thought, as you can imagine. And it is very interesting because, first of all, what I thought was interesting, I mean, what we tried to do at Skirball, there are two aspects. One was to really concentrate on basic mechanisms, so fundamental biology, discovery science, investigator driven science, curiosity driven science. So I think that is the principle. And despite the fact people may say, oh, there are so many different, we were just talking about that, areas of research, we all agreed, every one of us is interested in fundamental mechanisms. So there was a common denominator, but there was this diversity. And that is something that's really, really important because I do feel that creativity is rooted at the end in the individual. And that's where the investigator driven science is important to support. And that's also how I feel about the whole community. I feel like protecting people that they can follow their ideas. You want to have them in an environment where they're respected, where there are resources that they can do experiments, and where the collegiality is such that there's actually a lot of exchange. So a lot of what happened at Skirball was people were sharing ideas. And that is really important. And so it's interesting. On the one hand, I'm stressing the investigator driven research. On the other hand, I'm also really emphasizing the collegiality and being actually free in communicating with each other. So I think it's, again, those two parts that I see are really important for being creative, which is on the one hand, you have your own ideas, you're observing. And then on the other hand, the listening, where you're getting feedback. But you're getting feedback in a non-competitive way. You're getting feedback where people are actually taking their time to think about your project. And I think creating that environment was really, really important when we started the Skirball Institute. And I think it really worked very well. And so that is a spirit of empowering everybody, but really this community where people talk about science. And maybe because we're so diverse, there is also less reason to be competitive because it's not like two groups are working on the same project, for example, right?
So you explained some really important things that you did to give scientists the right kind of environment to be creative. But that sounds, in some ways, a little bit passive. I mean, it's certainly extremely important to give people the right environment. But would you say that is all you can do? Or are there ways in which to more actively bring scientists together or encourage them to collaborate, for example?
I think they should collaborate if they want to collaborate.
Yes.
And that should be natural. I think the most important is to hire the right people. When we were hiring people, we were hiring them not just being the most outstanding scientists, that was number one, but then just close after that came, and that was made very, very clear in the interview, we are a community. And so you're not going to be around, lots of neuroscientists, lots of cancer biologists, lots of immunologists. You're going to be around all these people who do all these different things. Is this something that interests you? And not everybody, some people feel much more comfortable to be in more with their same. And so we were selecting people not just on their scientific excellence, but also on this idea of being actually much, much more open to being in an environment where they're going to hear lots of different things. And I think that is being open to new ideas. That's part of the creativity. So that's what I thought was going to be more actively searching for people who probably were interested in their colleagues and were also interested in new ideas, right? And then another very important part was we really were looking for people who we really felt were also decent people, which is maybe the most difficult
Is that
one.
is that too much to ask?
It's hard to see that from the CV. But that's a combination. And what that leads to is that you have lots of discussions, and, through our PI meetings or the retreats, people would all of a sudden see like, oh my God, there's a really exciting connection we can make, right? So, for example, Dan Littman working with Wen Miao-gan on microglia, that was way before anybody else was doing this, and bringing the immune system and the nervous system together. So there were many examples of that, but it wasn't like, oh, we're doing this because our goal is to write a big grant. The goal was to bring together science and actually that's the creative process, to bring out new ideas by saying, oh, we should consider what immunologists find or approaches that immunologists use for our own work. So, for example, I think I was incredibly stimulated by Dan Littman's work on the microbiome to think about how do actually microbes in flies affect the biology of especially the germline. And we started to work on Wolbachia. And that was clearly, I don't know if I would have started to work on Wolbachia if it wasn't of listening to the immunologists' talks. And so I think that this is creating this environment. But I do think that it has to be scientist driven.
I think all our listeners, when they hear you explain, new collaborations are formed in the institutes that you lead, must find it so exciting as we do. But I want to ask you about something that you mentioned, which was that each person needs to learn whether they have it in them. And my question is it something you can learn? Do you have a kind of process where you see the trainees, the PhD students, and the postdocs, they learn to be creative? You see them maybe not joining your lab this way, but over time, as when they observe the project, when they see it's more than just reading textbooks, but coming up with your own ideas, do you see it develop in them? Or do you think you either have it or you don't?
No, I think you definitely can. I mean, not learn it. It's almost like opening a window. Because when you think of, I remember from school education not really knowing what science was. That when you do an experiment, you are opening a completely new world. And is that exciting to you, wherever that world is going to lead you? And I had this crucial moment one time when somebody who left my lab to go and leave science for another very different kind of career. And she said to me, I really like to complete projects. And when she said that, I realized, yeah, this is not what most scientists are like. I'm still working on the same project I worked on as a graduate.
we start projects. We don't.
Yeah. So, of course, we're
Starting good.
completing a paper, right? But it's like
Right.
when we complete that paper, there's already like lots of new ideas. It's never finished. There may be people where that's just not a good fit. And I think that doesn't satisfy them. And you could see that as being a real problem, right? Because in a way, when you think about it, what? You never finished? How can you ever be, sitting down there in your armchair and say, oh, look at my work? No, my work is going to be in the future, right?
Yeah, but to me, that's also what makes it so much fun, right? Like,
Yeah, exactly.
It's finished and then
The
it's
unknown.
over.
But if you feel frightened by the unknown or uncomfortable, and different scientists push it in different directions. I think you guys are pretty extreme in how you see it. But
yes,
I'll take that as a compliment.
Yeah. So Ruth, I was wondering, you told us about how you organized the institutes that you lead in just constructing the right environment for people to develop and for people to be able to be creative. But at the same time, you agreed that there is something to be learned about creativity. And so for your own group, for the students that you work with, Is there anything beyond the right environment that you do to help them be creative?
Sometimes when they have an odd observation and they say, oh, by the way, I saw this, and then to encourage them to follow that. I think so with time, they kind of learn that.
Well, Ruth, we started out the conversation talking about Saturday afternoon experiments that test crazy, high risk, high payoff ideas. And then we got to the point where we realized that the hardest thing for us to imagine is you coming home from work one day and saying, well, my research is finished. I've explored everything I wanted to explore. That's the least Ruth thing one can imagine saying. And so I think it's been so exciting to explore with you all the counterintuitive aspects of science that as much as we like to research, our research is never done.
Yeah, exactly. And there's lots of, I was thinking about lots of stories I wanted to tell you about particular experiments, but maybe another time. But
Absolutely.
where just something that fascinates you, or these puzzles, right, which makes you stumble, and then all of a sudden, I think with your questions, I felt like I almost wanted to write this down because it's sometimes, then it becomes like normal, right? Because a puzzle, once you sort of get to solving it, it isn't a puzzle anymore. And so you forget that it at some point was a puzzle.
Yeah, once you discover the question, its history sort of gets erased.
Yeah.
Well, thank you so much
Thank
for your
you.
time, Ruth. It was
I
such
really
a
enjoyed
pleasure
it.
and an honor
Yeah.
to talk to you.
Thanks very
It
much.
was really great. Thank you.
Thank you so much. And I love your podcast, so keep doing it. Thank you.
Thank you, Ruth.
We will.